Hi there, this is Alex Lipka. I'm going to give the third part of this lecture series, which is called Applications of Applications of Quantitative Genetics, Part 3. Yes, I can read. Okay, so here's my presentation. So what we're going to cover today, accounting for large effect genes in genomic selection, and we'll do a case study. How well does genomic selection perform for traits that do not have a complex genetic architecture? That is, traits controlled by a smaller number of genes. And then I'm going to talk about something new that I created specifically for this webinar series, which is using GS to indirectly quantify contributions of small effect genes. So a review of genomic selection, right? So what we want to do is we want to predict, well, breeding values using markers distributed throughout the genome, right? So we'll start off with a training population that are genotyped and phenotyped with the same markers as the breeding material. We'll use that to train a genomic selection model. Then we got breeding material that are genotyped with, again, the same markers. And then we plug in the genotypic values into our genomic selection model. We'll calculate genomic estimated breeding values and use that to make selections. So genomic selection is important because it enables selection without phenotyping individuals, and it was developed to speed up breeding cycles. So you get greater genetic gain per unit time, right? So various frequentist and Bayesian statistical models are commonly used and most produce approximately the same predictive abilities. Um, so I'm going to dive into one particular genomic selection model called RR-BLUP. And here it is. We have our phenotype of a white individual over here, and then every single marker throughout the genome is included as a random effect. And then we have our error term and our intercept. And then the BLUPs of these marker effects are subjected to this ridge regression penalty. So the thing to focus on for this penalty is that all predicted marker values are equally penalized so that they are shrunk to zero. So here's a picture of my first PhD student, and this is a thing that he studied for his master's degree. So I'm putting professor there because he's now currently an assistant professor at the University of Nebraska. So he was wondering, Professor Rice, that is, suppose we have a complex trait, right? So we got hundreds, thousands, tens of thousands of small effect genes, right, contributing to the trait. What if this complex trait had several large effect genes? Is the penalty I showed you in the previous slide, is this resulting in a substantial underestimation of the effect sizes of these genes? And are we unfairly getting bad predictions because we're not accounting for these large effect genes enough? So the random effects that I showed in the previous slide have the ridge regression penalty applied, but fixed effect markers do not. So, Professor Rice's hypothesis was that genomic selection models can make more accurate predictions if they account for large effect genes. And so the research question is, if we take that RR blood model that I showed a couple slides ago and add to it a fixed effect covariate of the peak GWAS markers identified in the training set, can we observe increased predictive ability? So we compared the predictive ability of the traditional RR blood model that I already introduced you to, as well as an RR blood model that includes fixed effect marker covariates. And we did a simulation study. And this is our simulation study. So we had the Goodman-Buckler diversity panel, and we had a sorghum diversity panel. I believe it's the SAP from Costa et al. 2008. We took these markers within each panel and we randomly selected them to be quantitative trait nucleotides. Okay. And then we simulated two different kinds of traits. Type 1, the QTN sizes were simulated as geometric series, so the effect size got progressively smaller and smaller and smaller. And then the second genetic architecture considered was where you had one large effect QTNs and then the remaining markers followed geometric series. And the user selected the heritability. So the way we evaluated predictive ability was through five-fold cross-validation. So you have a training set, four-fifths of your, either maize panel or sorghum panel. That was a training set. You ran a GWAS in it, and then you fitted an R-BLUT model that would include that peak associated GWAS SNP. Okay. And then you evaluated predictive ability in the validation set, and we repeated this 50 times. So going into this study, I thought that including these peak associated GWAS markers, you see a substantial increase in predictive ability. And I was wrong. Let me show you how wrong I was. Here's a picture of results. Now, our rows, okay, we have our results for data simulated in maize at the top. We have our results simulated for sorghum in the bottom row. And then we have the geometric series kind of trait. So a trait simulated without large, one single large effect gene. And then here we have the type of traits where we have one large effect gene, everything else is small. Now, on our x-axis for each of these plots, it's the number of markers that were included as fixed effect covariates. And then the y-axis was mean increase in predictive ability of including those fixed effect covariates in the model. And just to give you an idea of how poorly including these fixed effect covariates actually work, these black lines show a y coordinate of 0. So most of the time, like these box plots tend to be less than 0. So more often than not, we see a decrease in predictive ability when including peak GWAS markers as a fixed effect covariate. So usually it does a worse job than a standard R or blood model. And it gets even better. And by better, I mean worse. So here's one particular simulation scenario. We see a substantial increase in variability. So let's check this out. Our x-axis is number of fixed effect covariates included in our model, like number of GWAS signals, and then our y-axis is predictive ability. So here's an x-coordinate of 0. So this is the distribution of predictive abilities, okay, where we had 0 fixed effect covariates. And when we started including fixed effect covariates, look at how much greater the variability we see in these box plots. So this is telling us including fixed effect covariates increases the variability of predictive ability substantially. And it gets, and it gets even better than that, right? So one type of way to assess performance of a genomic prediction model is so-called checking your bias, right? So you will plot your observed trait value against your GEBV from genomic selection, and then you will see if and then you fit a simple linear regression model. So if your genomic prediction model is behaving correctly and not producing a lot of bias, I would expect to see my ordinary least squares estimate of my intercept to be around 0, and then my ordinary least squares estimate of my slope to be around 1. So check this out. Um, let's look at these slope estimates. So over here we have 0. Come on, mouse. So when we have 0 fixed effect covariates, we see that the slope is kind of close to 1. But then when we include um, a fixed effect covariate, it jumps up from something close to 1, which it should be, to something much higher than 1. And then we see when we subsequently add more and more and more fixed effect covariates, it starts to go down towards 1. But the take home is that since we're seeing this jump from approximately 1 to something greater than 1, we're seeing an induction of bias in our genomic estimating breeding values when we include fixed effect covariates in the model. So here's our conclusion. If you want to use genomic prediction, the risks of including these fixed effect covariates might outweigh the benefits. So what we suggest doing is conducting a cross-validation study, okay, where you evaluate the predictive ability, performance, with an increasing number of fixed effect covariates. Worst case scenario, you'll find out that you should not include any fixed effect covariates from GWAS in your model, and then you'll use the good old tried and true RR-BLEP-GS model. Okay. Um, Mind you, one quick comment. So if you're using a Van Raden kinship matrix, okay, your GBLUP model, which I'm going to show later, and you'll probably see a lot in the in the literature, and the RR-BLUP model, they're roughly equivalent, okay? Um, so now let's talk about the scenario where we run genomic selection on a trait that is not complex. And carotenoids, which are compounds, which here we're looking at them inside of maize kernels. Okay, um, they have provitamin A activity, and that's why we're interested in them. That's why I studied them when I was a postdoc, and it is established in the literature that carotenoids are not very complex. So they are controlled by relatively small number of genes. Okay. So here we had a 281 member Goodman diversity panel grown at Purdue University in the United States for 2 years. And then um, carotenoids inside of the kernels were quantified. Now, um, on Monday, when I talked, I was asked what is a minimum number for, a minimum sample size for GWAS, and I said something around 500. Here is a extreme scenario where I'm going much smaller than 500. Okay, so we wanted to look at a subset of this panel that had only um, uh, kernel color ranging from light yellow to dark orange because white kernels do not produce measurable carotenoids. Okay, so we got away with analyzing such a small sample size because the genetic architecture of carotenoids consists of really small number of genes. So in practice, I recommend analyzing a much larger data set than 201. That's what I'm trying to say. So this is a genomic selection study. So we were interested in comparing the predictive ability between a genome-wide set of markers versus one reduced set of markers. So here is the carotenoid biosynthetic pathway, and here are some related pathway enzymes. Okay, so we took, the resulting 58 candidate genes that have been identified once again in prior studies, and we considered only markers that were near these genes, and I forgot to activate this animation, but we also looked at this group. So we ended up looking at, 7,408 SNPs and 7 indels within the vicinity of these particular 58 genes. And then we looked at an extremely small set of markers where we only considered markers in LD with the genes producing these enzymes that are circled. And that resulted in 944 SNPs and 7 indels. Okay. So what we ended up doing was comparing the predictive ability from models that included, let's see, all markers across the genome, which we see in this plot over here. Okay, our x-axis is our particular compound that we had as a y variable in genomic prediction, and then our y-axis was predictive abilities. So the taller the bar, the taller the predictive abilities. And then we looked at markers near the 58 candidate genes, which similar plot is presented here in the middle, and then markers near these 8 QTL from previous studies, which are presented in this leftmost bar plot. And what do we see here? Well, we see that if we look across each of the plots, um, the box plots all look approximately the same. So this is telling us we're getting similar predictive ability whether we use a genome-wide set of markers versus an intermediate size set of markers near 58 candidate genes versus markers near just these 8 QTL. So in other words, models with markers near 8 QTL were as effective um, as genome-wide markers for predicting breeding values of these carotenoid traits. So the take-home message is that genomic prediction is a really valuable tool that has had an immense impact on breeding and should still be used. But the lesson here is that if you have a simpler trait, okay, you don't, you know, you will get similar predictive abilities by just focusing on the small number of genes that control the trait versus a genome-wide set. Okay, so now here's a new set of slides. Okay, so um, let me motivate this. So we can use GS indirectly to find evidence of smaller effect signals that may not be detected from a GWAS. So let me go to this. So GWAS, remember, the purpose of GWAS is to find regions of the genome with strong statistical associations with your trait. However, the problem with the GWAS is missing heritability. So GWAS typically finds only the largest effect signals to be statistically significant. And this means that, if you have a complex trait with thousands, hundreds of thousands of small effect genes, their true effect size might be too small to pass that threshold of statistical significance. Okay? So here's a study I'm going to show you where I contributed as a middle co-author, but we used genomic selection. Okay. So remember, genomic selection is for prediction. It's to find genomic estimating breeding values, right? So we're using that. We're utilizing genomic selection to indirectly tell us something about the contributions of these smaller effect genes. So essentially what we did was this. We compared two different genomic selection models. Okay. One of them accounted for putative a priori candidate genes. The other one did not. And we compared the predictive abilities of them. Okay. And so the standard GS model was model one. It's a GPUP model. I will explain it in a second. And then the second model accounts for genes with a priori evidence for the contribution to the trait. Um, so this is the paper, Turner Hissong et al. 2020. And what we looked at here was free amino acid content in seeds of Arabidopsis. So here's a picture of Arabidopsis. Here's a mock-up of the 5 chromosomes in Arabidopsis. And let's pretend that these orange dots correspond to genes that have been known to contribute to free amino acids in previous studies. So what we'll do is this. We got a genome-wide set of markers. We'll subdivide them into two different categories or into two different bins, if you will. Bin 1 contains all markers near these orange dots, near these genes known to contribute to free amino acids. And then bin 2 is all remaining markers throughout the genome. So um, the way we account for, these orange dots is we would fit this multi-bleb genomic selection model. This slide was created by my postdoc, Dr. Sarah Richards. So YIJ, this is the observed value of free amino acid in Arabidopsis seeds. We have a grand mean. Then we have this polygenic random effect, which follows this multivariate normal distribution. And this G1 matrix, okay? This is a Van Weyden 2008 relatedness matrix, okay? And it was calculated only using markers that were near, these genes depicted by orange dots. Okay. And then this G2I, this second polygenic effect, is almost identical to this, except in G2 over here, this relatedness matrix was calculated from all remaining markers that were not near that orange dot. Then, of course, we had our error term. So we have this model that accounts for the a priori genes depicted as orange dots. And we compare it to a standard GBLUP model, okay, where we have Yij as defined in the previous slide, have a grand mean. And then we have this polygenic effect, which just uses all markers throughout the genome, regardless of whether they're near the blue dots or not, okay? So, which does not account for those a priori candidate genes. And then we have our error term. So here is a summary of our results, all right? So we looked at free amino acids in Arabidopsis, okay? And each of these circles represents a different set of candidate genes, okay? So each circle represents a different set of orange dots on the Arabidopsis genome, okay? So for each of these circles, we compared a multi-BLUP model with a G1 and G2 that partitions markers along the lines that I've shown previously. It compares it and it compares it to a standard GBLUP model. Okay? So our x-axis, this is the extent to which the predictive ability of our multi-BLUP model that accounts for these genes is better than the predictive ability from a standard GWAS model. So the further out we go along the x-axis, the better of a job accounting for the corresponding genes does in improving predictive ability. And then our y-axis, this is the proportion of heritability of your trait that is explained by markers that are near these genes. Okay. Now, again, each circle depicts a different pathway tested using, this multi-BLUP approach. And then all of the circles that are colored in blue are statistically significant. These are statistically significant, better predictive abilities than a standard GBLUP model. Okay. So I put forth that all of the pathways that are colored in blue have evidence that the underlying a priori candidate genes are contributing to the genetic architecture of free amino acids. So the SNPs near pathways in blue explain significantly greater amount of trait variability than the same number of randomly selected SNPs. Okay, so what did we do? We considered the impact of genetic architecture on the performance of genomic selection. I showed an approach on how to use GWAS to indirectly help us find marker trait associations that GWAS cannot identify. And I think all of this is really cool because it goes beyond a typical genomic selection analysis. And what we're going to do next time is do that GAPIT demo for GWAS and then showing a demo for genomic selection using our RBLUP. So I'd like to thank you for your time, especially with watching this additional bonus lecture. I'm happy to answer any more questions that you might have. All right. Well, thank you, and hopefully I'll look forward to seeing you all on the webinar and in Cape Town in October. Bye-bye.